All right, so we're going to go a little more in depth in our PCR that we talked about yesterday. So first, let's just talk a little bit more about what goes into PCR reaction. So there's a thing called master mix. So we can order components for our PCR reactions, and those components can be called master mix. And these are available from commercial labs. So I have my template DNA. So the template DNA is a DNA that includes the target that I want to amplify. Everything else over here is going to be part of my master mix. So in my master mix, I'm going to have my DNTPs. Those are my ACT and G triphosphates. I'm going to have my PCR buffer, because remember I want to maintain pH. So this will maintain pH. Sorry, I lost my pen there. Use that to maintain pH. My magnesium chloride. Actually, this works as a cofactor. For DNA polymerase. It helps it work a little better. Helps stabilize DNA. I need my forward and reverse primers we talked about. And then my DNA polymerase and the type that we use in our lab is Taq polymerase, right? That heat staple DNA polymerase from Thermus aquaticus. So all that will be in my master mix, and I can order that together along with my forward and reverse primers, and it will come in a little microcentrifuge tube, and then I'll be able to do it. Okay. So all of that will go into my little mini microcentrifuge tube that's going to go into my PCR machine. All right, so DNA type is important as well. So remember, we talked about that PCR is really, really good for amplifying small pieces of DNA. Well, the problem is all of your DNA is what we call gDNA, or genomic, right? So gDNA is going to be that genomic. And it contains those introns. And the introns, remember, are NTRs, right? So this is a type of NTR, a non-translated region. So it just makes DNA bigger, right? We don't want big DNA, we want small DNA. So we would like to use, if all possible, cDNA. So if all possible, we'd like to use cDNA, right? Because cDNA comes from cDNA libraries, or complementary DNA. So this complementary DNA is DNA without the introns, right? So I can get that two ways. I can get that from growing bacteria in plasmids, right? So I can do that because bacteria don't have introns, or I can create using reverse transcriptase, right? So that's an enzyme that will take mRNA. So this makes cDNA from mRNA. Right, and remember mRNA is that transcript of your DNA that we use to make proteins, right? So usually we can do that, then we can store this. Once we do this, we can store it in what are called cDNA libraries, which are essentially plasmid libraries, right? Which are a storage vector. All right, so once we get our master mix, right, and we have our template DNA that we want to use, we have to figure out the PCR type. So in our lab, because I'm not super well funded, right, we have conventional PCR. So conventional PCR is kind of what we talked about yesterday. It runs on an algorithm, and we run 35 cycles, right? So we get up to that plateau. There we go. So we run 35 cycles in ours, right? And that gets us right to that plateau. Real-time PCR. Right, so real-time PCR, sometimes called qPCR, right, for quantitative, right? So that's right here. So real-time has photosensors and it measures photofluorescence. So what it does is it measures the amount of product, right? So it allows modification based on quantification. So the Q stands for quantification. So with the qPCR, right? So I can modify the cycles based on the product. So if I'm going to use this for diagnostics, right? So qPCR is good for diagnostics. I lost my pen again. So great for diagnostics. And diagnostics means diagnosing. So if I'm looking for a type of bacteria and I'm looking for DNA in a bacteria that may indicate a certain type of infection, then I can use qPCR, right? And if I get to a threshold where I'm confident that I have enough of that bacterial DNA, I can shut it off and say this is a positive result. If I run it for 35 cycles and I never get that flag, then it's a negative result. Okay, so qPCR is really good for doing something like that. So we have RT-PCR, so make sure you know all these abbreviations. So we have RT-PCR for reverse transcriptase. Remember we just talked about reverse transcriptase. We use it for making cDNA from mRNA. So this uses viral reverse transcriptase to make cDNA from mRNA, right? So we run that first at 42 degrees Celsius. Remember, because things like bacteria will live, in a temperature range between 35 and 42. So this enzyme will run at optimal running temperature for the bacteria that we got the reverse transcriptase from, or the virus that we got it from. And then we can run conventional or even qPCR after that, right? So we do a cycle at 42. So the RT is at 42. And from that I get my cDNA. And then we go up to 94 degrees Celsius for denaturation. We go to 55 degrees Celsius for annealing. We go to 72 degrees Celsius for extension. And then we keep going with that. So that's our thermocycler right there. Right? Denaturation, annealing, extension. And then I keep going down. But I need to do one round, right, with the reverse transcriptase. And we can use that to diagnose viruses such as influenza and COVID. So RT-PCR is how we diagnose COVID-19, right? And that's the gold standard for diagnosing. There's other types. Multiplex is another type. This uses multiple primers when I have multiple targets. So if I'm looking to see if something's been done, like if I'm looking for certifying food as non-GMO, there's a couple different vectors we use for inserting genes, and I might use primers for each one of those vectors, right? And if it comes up with any one of the multiple vectors that we use for making GMO foods, then we can certify it as GMO. Right? And if nothing comes up, then we can certify it as non-GMO. There's Degenerative Pcr is another one. Again, we use multiple primers. And we do that when the target DNA is not known, right? So if we know that we're looking for some sort of sequence, right? I might use multiple primers to see if I get a hit on it. This is usually followed. Right, once I get amplification, right, and my DNA is better known, then I can follow it with conventional DNA. We get nested PCR, right? Make sure you know that that's when DNA is suboptimal, right? So if I have a crime scene and I've got DNA that's been degraded, right? So what it does is it looks to kind of rebuild that template using multiple different primers as well, right? So second round uses additional primer to search for desired amplifier, right? And then we have fast PCR, right? So we use a different heating and cooling matrix, right, to reduce the time of 3 cycles. Usually takes me in my lab about 2.5 hours, but with fast PCR I could do it in 1.5 hours. So that would be like our rapid PCR testing. Okay, so not a lot today, but enough to keep you busy. All right, I'll talk to you soon.